Assistant Professor of Department of Mathematics, CVV-IST. I welcome Arya Chandran for the introduction.
Thank you, Sharat sir. Good morning, everyone. Welcome you all to the International Conference on Recent Advances in Composite Materials 2024, proudly organized by Chinmaya Vishwavidyapith, deemed to be university. We are excited to have such an enthusiastic and diverse group of participants gathered here to explore and discuss the latest advancements in composite materials. To ensure a seamless experience during the session, we kindly request your cooperation with the following guidelines. Please keep your mic muted throughout the presentation to avoid any interruptions. We encourage you to actively listen to the sessions and note down any questions that you may have. If you have any queries during the presentation, please type your questions in the chat box and the speaker will address them during the Q&A session. Let us all maintain professional decorum by refraining from interrupting the speaker during their talk. Thank you for your attention and cooperation. We look forward to an inspiring and thoughtful session ahead. Now, it is my distinct honor to introduce our esteemed keynote speaker, Dr. Augustine. He is a distinguished research scientist with extensive expertise in biomaterials, regenerative medicine, and tissue engineering, currently contributing his talents at School of Medicine, Stanford University as a research scientist. He earned his PhD in nanoscience and nanotechnology from Mahatma Gandhi University in 2015, following his MSc in bioengineering from Madurai Kamaraj University. Over the past 8 years, Dr. Augustine has gained postdoctoral experience across leading institutions worldwide, including Stanford University, the University of Massachusetts Lowell, Qatar University, the National Institute of Technology Calicut, and Technion Israel Institute of Technology. With over a decade of research experience, Dr. Augustine has pioneered advancements in bioengineered platform for trauma care, bone and skin regeneration, and pancreatic islet transplantation. His impactful research had led to the development of smart, biodegradable materials that improve tissue repair and function restoration, particularly in critical areas like military trauma and diabetics treatments. Dr. Augustine's work has been globally recognized with a ranking of 50th in biomedical engineering based on the Scopus Citation Index, along with inclusion among the world's top 2% scientists from 2020 to 2024. In 2021, he was honored with the Outstanding Researcher Award from Qatar University in recognition of his contribution to the field. He has also fostered a network of international collaborations across the USA, Europe, the Middle East, and Asia. Additionally, Dr. Augustine is dedicated to education and mentorship, training students in advanced research techniques and fostering an inclusive environment for emerging scientists. In this conference, Dr. Augustine will share insights on the evolving challenges and transformative potential of tissue engineering, focusing on skin, bone, and pancreatic tissue regeneration. We look forward to his thought-provoking discussion on this innovative solution reshaping the future of biomedical research and patient care. Please join me in giving a warm welcome to Dr. Robin Augustine. Over to you, sir.
Thank you, Dr. Arya, for the nice introduction. So, may I start now?
Yes, sir.
Hello.
Sir?
Yes, it's very clear, Dr. Robin.
Okay. Just a moment. Good morning. Thank you for the invitation to join with you today. So, I'm going to present something related to tissue engineering, the exciting field of tissue engineering. The tissue engineering, the challenges and promising. So I'm a research scientist in Stanford University, as the person who introduced mentioned, and I'm not going to again introduce. So, but a few words about Stanford University, where I am working now. Stanford University is established in 1891 by Leland Stanford, and it's a huge campus, 8,000-acre campus, which has several departments from medicine, engineering, law, a lot, and it's ranked in various ranking from it was ranked first a few years back by QS ranking in the world, but now it's ranked, I think, 3 or 4. So we have a lot of exciting research going on here, especially the innovative research that transform the daily lives of human beings. There are several companies and startup companies started from the research and academic activities or innovation activities going on in the Stanford campus. Some of the examples are the HP company. HP company started as a result of the motivation from the Stanford University campus. Actually, Stanford management that, and there are several industries, most two big industries here. Even the Tesla one of the main industries working at a walking distance from my office. And I am working in the we have several faculty members, including 1,100 faculty members, and around 17,000 students studying here. And I'm working in the medicine department, Stanford Medicine, especially in the department of radiology. So we have several sort of research going on. Most are related to the radiology related, molecular imaging, drug delivery, kind of research. And mostly I am focusing on tissue regeneration related research. For instance, the how the ultrasound can be used to treat diseases, how we can use ultrasound, therapeutic ultrasound, to regenerate the tissue. These kind of things are some of the aspects, but I'm not covering all of these kind of research today because of my limitations to tell all these, because many of these research are federally funded and supported by some industries, and I cannot disclose most of such research in public platform, especially when it is recorded. But generally, I will discuss what is tissue engineering. Tissue engineering is an interdisciplinary field that applies the principles of engineering and life sciences towards the development of biological substitutes that restore, maintain, improve tissue function or the whole organ. This is a famous definition of tissue engineering that is proposed by the fathers of tissue engineering, Joseph Langer and Wakandi. So simply, tissue engineering is the creation of tissues artificially in the laboratory by using human cells. That is, from the patient, we isolate cells depending upon the requirement. If we are planning to do some skin-related tissue engineering, we can isolate cells from the human skin and culture them in vitro. In vitro means in the cell culture systems, we can culture to get enough large number of cells to be transplanted. Then we can culture these on three-dimensional structures called scaffolds. Scaffolds are biodegradable porous structures, which I will explain later, how we fabricate these and all. Then this kind of structure, porous structure, we seed the cells in the presence of growth factors, growth factors and other nutrients. We culture to proliferate well and get a tissue, an engineered tissue. Then after getting that, we implant into the patient back. we can also use stem cells and differentiate stem cells into required types of tissue or cells or tissue, then implant back. This is the basic concept. Why this tissue engineering is important? Because we need enormous or large quantities of tissues because of the road accidents, sports injuries, burn injuries, and even plastic or cosmetic tissue, cosmetic surgery tissue that is very difficult from patients of body or from other patients. And there are several other complications, kind of like immune response, immune rejection, everything, if we get the tissue from another donor. And also we can create several other types of tissues, for example, heart valve and liver transplant, then pancreas transplant. I mean, we are not creating pancreas as such, but we can make a engineered platform where pancreatic islet can be seeded and delivered. Even for cornea tissue regeneration purposes, we can use tissue engineering. And there are several components of tissue for tissue engineering. So the porous structure that support the cells. It can be natural or synthetic in nature. And we should have cells that can be mesenchymal stem cells, for example, or other cells like osteoblasts, bone-derived cells, or skin cells, or whatever as per the requirement of the project. Then we should have biomolecular cues that help in tissue regeneration, for example, growth factors or nutrients or amino acids. These kind of things can be incorporated into the scaffolds, which will support the tissue regeneration process. So what are tissue engineering scaffolds? These are something like a support material. For example, when we construct buildings, I hope in the what's going on with the scaffolds and structures, supporting structures, because it's a new university and so many construction works going on, something like that. Similarly, the scaffolds, bio scaffolds are also like a support, temporary support platforms that can support the human cells to grow and to proliferate well. So these should be able to encourage cells to grow through it. Then there should be enough porosity and permeability to allow the nutrients to reach everywhere in the structure so that the cells will grow well. That's it should be highly porous in nature. Then these scaffolds should be biocompatible in all respects. That means it should be non-toxic, non-carcinogenic, non-teratogenic. Non-teratogenicity means it should not cause any abnormalities in the fetus if it is implanted in a pregnant woman. So, and also it should be biodegradable and bioabsorbable in nature. And there are several instances where we use non-biodegradable scaffolds also. For example, in the islet transplantation research, we use non-biodegradable scaffolds. And the key features of the scaffolds used for tissue engineering are morphologically, it should be able to mimic the extracellular matrix. That is the support material naturally present in the human tissue is like collagen kind of material, fibronectin kind of materials are present in the body. That kind of structure should be mimicked by the scaffolds. Then the physico-mechanical properties, hydrophilicity, stiffness, strength, degradation rate, also should match with the natural extracellular matrix. So then also it should be able to support cell migration, cell adhesion, cell proliferation, etc., as in the biological natural extracellular matrix. Then we can use several natural extracellular matrix kind of materials also, like glycosaminoglycans, fibronectin, hydroxyapatite, hyaluron, and collagen, etc., to functionalize the scaffolds to get more biological response or ECM mimetic properties. And conventionally, we use several synthetic, natural, and natural synthetic hybrid materials for tissue engineering application. For example, synthetic materials like polycaprolactone, which is biodegradable, also polylactic acid. Lactic acid can be synthesized natural way as well, but also there are synthetic way of making it. So we can consider either it as a biopolymer, natural biopolymer, synthetic, based on the way we make. There are several other natural materials used in tissue engineering, like collagen, gelatin, pectin, alginate, so on. And also we can develop natural synthetic hybrid materials using natural polymers to improve the properties. Like, for example, if you want to control the degradation rate of gelatin, we can methacrylate material like gelatin methacrylate, which can be even photo cross-linked using UV light or light with optimal UV wavelength. Materials can be developed this way. There are several scaffold fabrication methods. For instance, we can use self-organization techniques like biomolecular organization approaches to make porous materials, and also casting techniques. We can make a porous porous mold. Then in this mold we can pour the polymer solution and make scaffolds. Then we can also incorporate various additives like porogens. Porogens can be salt-based porogens, and also like carbon dioxide generating kind of materials. Such materials can be used so that the carbon dioxide will be released during the processing. Then when the carbon dioxide is released from the polymer, within the polymer, the way the carbon dioxide move out will create porosities. There are several other techniques. For example, we can use electrospinning, 3D printing, 3D bioprinting, and so many things we can use to generate polymeric scaffolds. Even graphene, we are using graphene based scaffolds. There are several ways to make. The idea is that it should be highly porous in so that the cells can penetrate. I used electrospinning in several of my projects from my PhD. Even now I am using, because it's a very, very nice technology, although it's a bit conventional, but it's very good to make highly porous and highly flexible membranes for several tissue regeneration applications. These are very excellent for skin substitute application, wound dressing, making, etc. So technology uses high voltage power supply to drive polymeric solutions to a collector from a syringe pump. In a syringe, we take the polymer solution, apply high voltage like 15 kilovolt, and then due to this high voltage, the polymer will be charged, and when we apply some specific flow rate using a syringe pump, the droplets come out from the needle tip, will move towards the collector because of the electric charge inside the polymer. And we will collect this on the drum collector or in a flat collector, which should be a metallic plate or metallic drum, from where we can peel out and take like that. You can see like how flexible these materials are. And this is like the instrumentation, how our electrospinning unit. So yeah, you can see the electrospinning process here. So there is a syringe you can see here, and it's connected, and you can see it's working. I think, I don't know how easy to how visible the fibers, but the process is like that. Anyway, we are applying the flow rate, the voltage kind of things. Then yeah, then the electrospinning process is started. Yeah, now high voltage. Yeah, now you can, I don't know if you are able to see, but yeah, it's rotating now. Yeah, I think you are able to see, but I'm skipping this. Yeah, it looks like that. After the electrospinning process, whatever we got looks like that. It's very flexible, like a tissue paper. And we can use for skin regeneration application, or even I'm using this for islet, pancreatic islet transplantation kind of challenges in tissue engineering. Facilitate the vascularization process to achieve the vascular. So we will have human cells in the construct. So without a vasculature or blood vessels, without getting oxygen, the cells in the scaffolds will die. So that is a major problem. So we need to the blood vessel formation soon after transplanting or implanting the scaffold with the cells inside the body. So that is one big disadvantage, I mean, big challenge that we are trying to solve. And the maturation of cells into different types of cells, especially when we use stem cells for the tissue engineering, then the cell migration or cell infiltration inside the scaffold. Although the scaffold for us, it's not the scaffold structure. So we need to improve that. Then the infection at the implantation site, when we especially use the materials like natural materials, there is a chance of infection. So, and also scaling up large scale tissue engineering process is difficult to achieve because of several obstacles. Overcome such challenges include use of nanomaterials are signaling agents, growth factor, immobilization to improve the cell proliferation, etc. Then chemical function, for example, the cell attachment, conductive materials to provide stimuli, electrical stimuli for the cell proliferation. Also, to improve the degradation capacity of the scaffolds, we use several approaches like incorporation of nanomaterials. Then also we are trying to improve the mechanical properties of the scaffolds by nanomaterial incorporation kind of approaches. Then also we try to improve the structural features by incorporating various approaches. For example, modifying the polymers using several leaching agents. And also we use a graphene kind of material, CVD graphene, using novel approaches. Surface topography is an important factor that affects the proliferation of cells. So, for example, in a flat surface, the protein absorption is minimal or non-specific in nature. For example, when we modify such surfaces with nano scale features, the cell the protein absorption will be higher than cell adhesion, specificity, cell proliferation, cell differentiation, etc., will be maximized on the nano structured materials. We used nanofillets. Nanofillets are nanomaterials that are incorporated in the polymers to improve the properties, especially the tensile properties. We found that when we incorporated zinc oxide nanoparticles in the scaffolds, electrospun scaffolds, we found the improvement in mechanical properties at optimal concentration. The importance of such studies, such research is that we need to specifically incorporate or find the specific concentration of the nanomaterials used in the scaffold. For example, here 1% of nanomaterial seems optimal for the cell, enough mechanical property, while we incorporate 6% zinc oxide nanoparticle because of the agglomeration that you can see here, the properties decreased. Also, we try to improve the biodegradation of the scaffold. For example, in the case of polycaprolactone, it's a biodegradable polymer by hydrolysis. And such a hydrolysis process generate finally the acetyl coenzyme A, which is used in citric acid cycle. And we incorporated nanoparticles like zinc oxide to improve the degradation because of the specific properties that generate reactive oxygen species kind of things in the within the system. The zinc oxide nanoparticles and also the photo oxidation capacity kind of properties of zinc oxide nanoparticle, it can improve the degradation. When we incorporated around 4% zinc oxide nanoparticle, we got very high degradation of polymeric fibers, which is very important because optimizing the degradation of materials is very important. As the tissue, you can see in the right side, that is the hydrophilicity of the scaffolds increased over the time, especially when the zinc oxide nanoparticles are there, because the degradation of the scaffolds expose hydroxyl and carboxylate groups, which are hydrophilic in nature, and so the hydrophilicity of the material increased. So nanoparticles can be used as active ingredients in the scaffold as well. For example, we used several metal oxide nanoparticles, especially like zinc oxide, cerium oxide, titanium dioxide, yttrium oxide, several nanomaterials to improve the biological performance by using them as active agents. When incorporated in the polymeric scaffold, these were able to support cell proliferation, cell migration, angiogenesis. Angiogenesis means the blood vessel formation, and also provided antibacterial activity, especially the zinc oxide nanoparticles, ultimately resulting into the higher wound healing activity. For example, when we use metal oxide nanoparticle like zinc oxide or titanium dioxide, they are able to generate reactive oxygen species. Such a reactive oxygen species are redox active materials in the biological system. At optimal concentration, they can provide better cell proliferation, angiogenic response, and all. However, if the concentration is higher, excess reactive oxygen species can cause deleterious effects as well. So also we use the zinc oxide nanofibers kind of materials for higher endothelial cell migration and so on. Yeah, in another study, we used the antioxidant nanomaterial like cerium oxide, yttrium oxide, etc., which can act as nano enzymes or nanozymes to help in diabetic wound healing, because diabetic wounds are in excess oxidative stress, and providing the antioxidants or nano enzymes that can degrade oxidants will help in diabetic wound healing. So, for example, also we use nanofiber based materials as a protective coverage over the wounds, like a wound dressing, to prevent the microbial penetration into the wound site, which will help in prevention of the infection, while the fibers in the scaffold act as a cell migratory channel to help in fast wound healing. You can see that how we apply the wound dressing over the animals. These are guinea pigs. The scaffold or fibrous membrane over the animal, the wound generated on the animal, and then measured the wound healing over time using this formula. So we can see that when we use zinc oxide incorporated polymeric scaffolds, the wound healing was faster. On day 3 we can see day 30 we can clearly see that the wounds were completely healed when the zinc oxide nanoparticles were present in the scaffolds. Another important factor is the blood vessels. We are trying to increase the blood vessel formation, means the formation of blood vessels, which is very critical for research, which is very challenging as well. To ensure the nutrient and oxygen supply and waste removal, we need angiogenesis, that is the blood vessel formation. So otherwise, if without blood vessels, the implanted engineered tissue will die. So we use growth factors like VEGF or other growth factors to improve the blood vessel formation. So also there are several ways to study the blood vessel formation. One of the approaches, we use chicken chorioallantoic membrane assay for this purpose. So this is a relatively simple approach which we can do in our laboratories. We incubate the chicken egg, and on day 4 we remove the shell, then implant the scaffold. Then we again further incubate for 24 hours. Then remove that, we cover initially with some tape. Then we remove that tape and observe under microscope to see the blood vessel formation. Then, yeah, this is the approach we use. I think I need to skip because of the time limitation. So this is how we cut the eggshell. Then add the scaffold inside this. Then on the bottom corner you can see the blood vessels and embryo. Yeah, so we do this. And yes, when we tested such material, for example, PCl zinc oxide composite scaffold, we can see that the bare PCl won't initiate the new blood vessel formation, whereas the nanoparticle loaded scaffolds increase the blood vessel formation in the chicken chorioallantoic membrane. And we implanted such materials inside under the skin, that is subcutaneously in a guinea pig model. Then we found that large number of blood vessels are formed, which is very clear from the histology images which is shown in the bottom part. So also we use electrical stimulation based approaches to increase the cell proliferation, blood vessel formation, etc. by using piezoelectric scaffolds. For example, like piezoelectricity is the ability of materials to generate electricity upon the mechanical stimulation or mechanical while applying mechanical forces. And the vice versa phenomena also can happen in such materials. That is, when we apply electrical stimulation, the material will produce mechanical force, mechanical deformation also. So we use such material. For example, PVDF TRFA or PVDF is a piezoelectric material, which is such properties is due to the distribution of different phases, polar, that is the polarity of the material. So such polar materials can generate electrical response. So how we use such materials in tissue engineering is like the similar way. By electrospinning, we generated PVDF TRFE and the PVDF TRFE zinc oxide nanocomposite scaffolds implanted in the animal rat models subcutaneously. We also cultured several types of cells, mesenchymal stem cells, human umbilical cord endothelial cells on such scaffolds to see the cell proliferation. We can see that when zinc oxide nanoparticles were there, when implanted in the animal models, you can see in the histology images in the bottom corner on 7th day, we found several blood vessels. That red things that you are seeing here are the blood vessels and the RBCs. So we found a large number of blood vessels when zinc oxide nanoparticles were incorporated into the scaffolds. Also, we use similar way the titanium dioxide nanorods loaded scaffolds for tissue engineering, tissue regeneration. Because of the time limitation, I'm not going through in detail. Also, we use cerium oxide, as I mentioned, antioxidant nanoparticle in polyhydroxybutyrate valerate scaffolds. In the fibers, the nano ceria were loaded. Then in vitro and in vivo studies, in vivo studies were performed. In vivo means chicken embryos. Then we found that higher cell proliferation, cell adhesion, cell migration, and blood vessel formation, which resulted in higher wound healing activities. Yeah, you can see here, and when we conducted cell cultures, the cells were able to be attached very well on the scaffolds and provided higher cell migration when the cerium oxide was present, and also provide higher wound healing. Also, we used such nanomaterials, cerium oxide nanoparticles as nanoenzymes in gelatin methacrylate scaffold. GELMA means gelatin methacrylate. Then using a photo initiator, we cross-linked GELMA containing cerium oxide nanoparticle to make a hydrogel. Then after freeze drying, we got highly porous scaffolds. Because of the antioxidant property or free radical scavenging activity of cerium oxide nanoparticles, the ability to support the material to cell proliferation, etc., was much higher, and which helped in diabetic wound healing. And also we are doing the growth factor conjugation of scaffolds, especially by conjugating CTGF, that is connective tissue growth factor, in with reduced graphene oxide. Reduced graphene oxide was used as a carrier material, which facilitated the controlled release of the CTGF. Also, the graphene has good angiogenic response, graphene oxide. So we found that the cell proliferation, everything was higher when optimal concentration of the graphene oxide and also the CTGF was found present in the scaffold. Also, we found higher diabetic wound healing. Similar way, we used the CTGF in highly porous electrospun scaffolds with high porosity. Depending upon the porosity, we were able to control the release of the high or the growth factor, which also provided higher mechanical strength when we coated the PLA. The main material was the PLA, and also we used PVA for the the coating material. Then when swelling was very high when PVA was used, so optimal swelling is required for the wound healing and also for the release of the growth factors. Yeah, so this shows the proliferation, I mean the migration of different cells. I don't know whether you are able to see it clearly, the cells, but the 3T3 fibroblast, had keratinocyte, and the endothelial cells were seeded on place. Then when we made a scratch wound, that is a wound on the cell culture system, then kept the material over that, which containing CTGF, then the cells moved, I mean, proliferated or grown faster and healed the wound when the CTGF was present. Similarly, the higher blood vessel formation was observed when CTGF was there. Similarly, we generated a hydrogel, hybrid hydrogel patch. In this approach, what we did, we used GELMA, that is gelatin methacal oil, loaded with epidermal growth factor, which is a potent, very highly potent growth factor that can help in wound healing process. Then we loaded such material on the wound. Then we applied a secondary PHVV mesh, that is electrospun mesh, over that wound with hydrogel. Then we applied the ultrasound. Then what happened? That material, that hydrogel along with the porous scaffolds were integrated into the wound. Then we allowed for the wound healing, and we found that very high rate of wound healing was observed when the protective hybrid patch was over there in the wound. And also we developed a new approach, that is air jet spinning, using a high pressure pressurized air. We drive the polymer from a brush, air brush, to a collector, and then we collect the fiber in similar way like electrospinning. This is a relatively very new approach. We did a lot of optimization to achieve this, and in one of the studies which is published, you can refer this paper. Because of the time limit, I am not going in detail. You can refer this. This is an interesting approach. Everyone can try. So air jet spinning, using that approach, we developed such a material and used for tissue engineering application. Basically, we use polycaprolactam and diamond nanosheets. Diamond. This was conducted in collaboration with the Dr. Manish in NIT Calicut. He made these nano diamonds and provided us, and we made the scaffolds. So such collaborative work can do great things, which was very highly useful for tissue engineering. We also used 3D printing based approaches for bone tissue engineering application. You can see the proliferation of osteoblast, pre-osteoblast, over the 3D printed fibrous scaffolds. So this is how we do the 3D printing. Maybe you all know, because it's a common approach. And also we incorporate, as I mentioned, the blood vessel formation is a limiting factor in tissue engineering. So to achieve that, we use various approaches like growth factor incorporation and all. However, still it takes some time for the blood vessel to form in the implantation site. So we thought how incorporating the oxygen generating materials can help to overcome such a challenge. So we developed oxygen generating nanomaterials to incorporating tissue engineering scaffold to improve bone regeneration. So here what we did here is we synthesized calcium peroxide nanoparticles, calcium peroxide nanocuboids, which has a cube-like structure. Then this material can generate oxygen. First they produce hydrogen peroxide, then it will be converted into oxygen in the presence of catalase in the body. Then this oxygen, when we incorporated in electrospun scaffolds and used in hypoxic condition, without oxygen, we found that pre-osteoblast cells can proliferate very well and survive in hypoxic condition and help in bone regeneration. This was the basic approach using calcium chloride and ammonia. In the presence of PEG, we synthesized calcium peroxide nanocubes, which has a cube-like structure, and the SEM morphology shows that cube-like structure. It was like 35 to 200 nanometer in size. Then when we incorporated in electrospun scaffolds, the fiber structure was like that. We developed aligned fibers using a rotation mandrel. Then we characterized it using various techniques, which I'm not showing all of these. We also conducted mechanical studies, so many studies. Then we performed oxygen release studies without cells and with the cells, pre-osteoblast cells. We found that for up to 14 days, they were able to provide the oxygen in a sustained manner when we have various concentrations of oxygen generating material compared to the bare PCl material without any cells. So we can see that when we had the calcium peroxide oxygen generating material in the PCl scaffolds, the cells were proliferated well, especially when we see on this panel, like the PCl containing 3% nanoparticle and 2% nanomaterial, we can see the we can see here this one. Here you can see a lot of cells proliferating well. Even up to day 14, we can see here a lot of cells. The green ones are the living cells and the red ones are the dead cells. So green cells are good, dead cells are bad, right? So in the hypoxic conditions, without without the oxygen generating material, we can see almost all the cells are dead. That is this right side graph. The green one shows the living cells, and the red one, the bottom one, shows the dead cells. We can see the compared to the bare PCl membrane, more living cells were when PCl were incorporated with the oxygen generating material. However, at higher concentration, we observed a little decrease in the viability because of the slight toxic response, because the calcium peroxide can generate reactive oxygen species. So we need to use optimal level of materials, whatever case. Also in the Stanford University, as I mentioned, I have limitations to tell all these intellectual property related things. So however, we are doing pancreatic islet transplantation kind of research, which is very clinically relevant thing. We are also helping clinical teams to develop biomaterial scaffolds. In one of such studies, we may develop graphene scaffolds. This is pure graphene scaffold without any polymer, just graphene. We made as a highly porous structure using a special CVD approach, using a template. And then we see that that green colored thing that you can see is the pancreatic islets isolated from the mouse, which is growing very well after 5 days. Otherwise, it's very difficult to achieve such a proliferation without a bioscaffold. And we also do stem cell transplantation in the patients, and we develop like clinically relevant collagen based materials for the transplantation of pancreatic the mesenchymal stem cells derived from bone marrow, umbilical cord, and various sources to the patients. So for that we develop scaffolds. As a bioengineer, my duty is to develop such material and also test in vitro systems, and we have animal surgeons to perform animal experiments, and the clinical studies are performed by our group leader Avinash Thakur and team. However, we are closely involved in such research. And we are also doing 3D printing related research. That is, you all know about 3D printing. So we are doing 3D bioprinting, that is printing human tissues as such in the lab using bioinks. Instead of polymers, we use hydrogels containing cells and specific growth factors, nutrients, oxygen generating materials, etc. And then print this as organs or tissue and implant into the patient. This is the basic approach that we use. And let me conclude now. As I have mentioned, tissue engineering has a lot of potential in regenerative medicine, although there are challenges. So microporous and micro-patterned scaffolds can act as supports for cell proliferation in tissue engineering applications. Various nanofillies can provide mechanical property, can improve the mechanical property of the scaffolds. Incorporation of growth factors, nanomaterials, etc., can improve the functional property like cell proliferation, cell migration, and tissue regeneration. Then several nanomaterial can act as agents to promote cell adhesion, proliferation, angiogenesis. Same thing. The 3D printing and 3D bioprinting has a lot of potential in revolutionizing the tissue engineering, and let's hope this will be in the clinic within the next couple of years. And thank you for your kind attention. And also I thank the whole team of this who organized this conference. And I hope this kind of platforms, opportunities will come in future because of the great vision of Chinmaya Vishwavidyapeeth and all. And thank you, Dr. Praveen, my friend Dr. Praveen, for especially inviting me. And this is my email address. If any of you are interested to contact me to learn more about such techniques, and I also have some plan to start some trainings in India related to especially the 3D printing, then electrospinning, air jet spinning kind of approaches in India. So if you are interested to collaborate with me or to get some training or kind of things, let me know. You can email me. Thank you very much.
Thank you so much, sir, for such an insightful and thought-provoking keynote address. Your expertise and innovative perspectives have truly enriched our understanding of the critical area in material sciences. Now we will move on to the Q&A session. If you have any questions, please type in the chat box. Professor will address the questions one by one. Dr. Augustine, we have a question from Mr. Atul Ravi, and the question is, what are the major challenges of incorporate nanomaterials to a polymer scaffolds, especially biopolymers?
Yeah, thank you very much for this question. And yes, as I mentioned, one of the major challenges is the polymer property may be affected. For example, sometimes we need large quantity of nanomaterial to get optimal mechanical property, I mean optimal biological performance sometimes. In such situation, for example, antimicrobial activity, when we use biopolymers like polycaprolactone or even hydrophobic materials, we need to incorporate maybe 10% of the nano material in the polymer. But the problem is that such a concentration may not be very suitable for in terms of the material property. So it may compromise the material property. For example, the mechanical property may be affected because of such a higher concentration, and also the crystallinity may be affected. So that is one problem. The other problem is sometimes to get certain property, for example, yes, antimicrobial property, we can take it as an example again. We need to use maybe higher concentration. Then in that concentration, that material may be toxic to the human cells. So we need to play with that and find, an optimal concentration of the nanomaterial without affecting the material property, without affecting the cell adhesion, cell proliferation, cytotoxicity, I mean the biocompatibility, etc., while providing a better or optimal functional property. That is the second challenge. And there are several other challenges. For example, the degradability and removal from the biological system will be a problem. And some of the materials are, although we are doing a lot of research, so detailed studies, when coming to the detailed studies like teratogenicity, for example, when it is implanted in a pregnant lady, or if it causes the abnormality in the fetus, that is a big problem. But such studies are limited because of our inability to conduct such detailed studies. So these are, and also environmental impact is there. We can incorporate nanomaterials, but these won't degrade very quickly in the environment. Silver nanoparticle, we are all using silver nanoparticle for getting antimicrobial property, but these materials won't degrade or won't lose its functionality in the environment. So it can kill useful bacteria, environmentally friendly bacteria, and cause even it can cause bioaccumulation, biomagnification, etc. in the animals. Even in the plants, it can be absorbed and it can come back to the human beings. So many things are there, and all these things we need to address. These are the challenges. Thank you.
Sir, we have one more question from Archana MS, and the question is, When we incorporate the nanoparticles in electrospinning techniques, it's necessary that the particles are biocompatible for scaffolds. Can we use biocompatible conducting polymers incorporate with the other polymers in electrospinning techniques for scaffolds formation, especially for bonds?
Yeah, the nano, for sure, the material that we use for the biomedical application, I mean, implantation application should be biocompatible. Whether it's irrespective of nanomaterials or polymers, everything should be biocompatible. Because even if we encapsulate some material inside the polymer, it will degrade over time, right? Then when released from such polymers from the polymer matrix, this will release. Then that can be absorbed by the cells. Then it can affect the cells and cause toxicity, and the patient will be in difficult over time. So we should definitely use biocompatible nanomaterial. And the second question, I think, was related to the polymer, like, yeah, we should use biocompatible polymers. Could you please repeat the second part of the question?
Yes, sir. Yes, sir.
Is something related to the polymer?
Yes, yes. Can we use biocompatible conducting polymers incorporate with other polymers in electrospinning techniques for scaffold formation, especially for bonds?
Yeah. Definitely, we can use conducting polymers, but the problem is that most of the conducting materials are non-biodegradable in nature, and even if we use such non-conducting polymers in the biodegradable polymers, these can remain in the body. But for example, when we use for instance, in the islet transplantation, where we need a long-term stability for the scaffold, because islet should be protected from the body, from the immune response, and also we use generally the highly non-degradable materials like PVDF. Then in such cases, definitely we can use conducting materials, and which is good for cell proliferation. And graphene is one of the, I mean, the best conducting material, right? We are using such material so that the cell communication will be very good. Thank you.
Thank you, sir. And one more question from Vidha. What is the advantage
Okay.
of using GELMA? As it is providing gel metrics, why specially GELMA used? Is there any pH requirement while using this?
Yeah, so gelma is gelatin methyl, which is a methylated form of gelatin, which has more stability than gelatin in terms of degradation, and all. We can control the degradation by adjusting the cross-linking intensity. Like when we are doing photo cross-linking approaches, we can tune it by controlling the wavelength, by providing different wavelengths of cross-linking light, and also cross-linking time can be varied, and also we can use various amounts of cross-linking agent, photo initiators, while when we are increasing the photo initiator concentration, the degradability will decrease, degradability will increase. The same way, the mechanical properties also can be controlled by increasing the photoinitiator concentration, which is not very easy in gelatin. Also, where we need photo cross-linking approach for several applications. For example, when I use such materials for that wound healing experiment, you may remember that the polymer placed over the wound and use the light. To get the light cross-linking, that is photo cross-linking activity, we need a gelatin, GELMA, that functional group. You can check in my papers for the specific details how the functional groups help in functional group, beta-acryloid functional group help in wound healing, help in the cross-linking. Thank you.
Thank you, sir. Thank you, sir. There is no further questions. We can wind up the session. Once again, thank you so much, sir. Your
Okay,
detailed
thank you.
response to Your detailed response to all the questions are much appreciated. We also extend our heartfelt gratitude to all the participants for their active involvement and thought-provoking questions. Thank you once again, sir, and have a great day ahead.
Thank you.
Thank you, Dr. Robin. And I know that the participants has many questions, but as
Yes.
due to this time restriction, definitely they can contact you. Your email you have provided, they can write to you later. And regarding plan you mentioned, definitely after this conference we can discuss the training kind of things. Definitely we can discuss.
Yeah, sure. You can contact me. Anyone can contact me or ask any questions. I'm very happy. Thank you very much.
Thank you. So dear all, the session now come to an end. We will start our next technical session by 11:35. I request everyone to take a quick break, and those who are going to present articles in the